Session?
Yes, yes, yes.
Okay, thank you very much. Dr. Asif sir, you can just share your screen and presentation. A very good afternoon to all the participants, including our respected resource person for today's lecture. Sir, your presentation is visible. Just for the information of all the participants, I just want to give a brief introduction of yours. Dr. Asif sir is one of my close friends and my senior also. He was from Downey University, Dr. Y.S. Parmar University. Dr. Asif has done doctorate in forestry sciences from Dr. Y.S. Parmar University of Horticulture and Forestry, Solon. Presently, he is working as assistant professor in Faculty of Forestry, Sher Kashmir University of Agricultural Sciences and Technology. And his area of research, as you might be saying the title of this talk, is by and large related to ecological analysis and conservation prioritization of forest habitats and other areas like grasslands, etc. And he will be delivering a talk. This topic, although it may not be directly related to agroforestry, but just like we had a lecture in the morning on on energetics of agroforestry systems, this is somewhat a new topic and a new dimension to agroforestry research because in agroforestry, as all of you know, it's a diversified system. There are several components that are integrated on the single platform. So obviously, we need some ecological analysis so that all the components are have that ecological setting in mind because that is something which is very important so far as our traditional agroforestry systems, especially home gardens. As you know, there was a lecture on home gardens. They are very diverse ecosystems. So I welcome you on behalf of all the organizers, NADCL, ICAR, IGFRI, Regional Research Station Srinagar, and on my personal behalf as well. I welcome you, sir, for sparing your time for this talk, for this lecture. The floor is all yours. I think 50 participants have already joined. You can start your presentation. The floor is yours.
Uh,
I urge all the participants to have their queries in the chat box or they can directly interact after the presentation is over.
thank you, Rafael. Am I audible?
Yes, yes, sir. You are audible. The slides are also visible.
Okay, thank you. Okay, thank you, dear. A very good afternoon to all. I am Dr. Asif Ligotu, working as assistant professor at Faculty of Forestry at SCOS, Kashmir. Today's topic is ecological analysis and conservation prioritization with some practical aspects. To start with, let us have a definition of a population. What is a population? Population is generally a group of individuals of a particular species occupying a particular geographical area at a specific time, which includes monocultures like agriculture, plantations, etc. While as on the other side, a group of several species, plants and or animals living together with mutual tolerance and beneficial interactions in a natural area is known as a community, and more appropriate, it is a biotic community, which include systems like agroforestry, etc. And these systems, which are community systems, are much more complex than a monocultural systems like agriculture and plantation. The community ecology aspects, basically, there are two aspects in a community ecology. One is the structural aspect, and the other aspect is the functional aspect. And the community ecology deals with the characters that describe the structure of a community and the system of the classification of communities and the process of community dynamics. And in a community, organisms share the same habitat growing in a uniform environment. A forest, grassland, desert, or a pond, these are the natural communities, while as the systems like agroforestry, these are the artificial ones. A community must include only living entities of the area. If we include non-living or abiotic factors together with the living entities or biotic factors, if both are considered, then we are dealing with an ecosystem rather than a community. And like a population, a community too has its own characters, which are not shown by an individual species or individual population. These characters, which have meeting only with the reference to a community level of organization, and these include species diversity. In species diversity, each community is made up of different type of organisms, be it plants, animals, which are taxonomically different and are differ from each other. The number of species and population abundance in community varies greatly. And the second character is the growth form and the structure. Community is described in terms of major growth forms such as trees, shrubs, herbs, etc. And each growth form, as in case of trees, there can be different type of plants as broadleaf trees, evergreen trees, and according to the mode of arrangement of these various growth forms, the community exhibits different layers. If the layering is in the horizontal side, we call it as zonation, and if it is a vertical layering, then we call it as a stratification. There another character is a dominance. In each community, all species or all populations are not equally important. There are relatively only a few of these which determine the nature of that community. These few species exert a major controlling influence on that community, and such species or such populations are known as dominants in that community. And regarding the functional aspect of a community, there is some characters like succession. Each community has its own developmental history. It develops as a result of directional change in time in it with time. And the second is the trophical structure. Nutritionally, each community exhibits a self-sufficient, perfectly balanced assemblage of the organisms. Now, dealing with the phytosociological studies, surveys are important tools to ecologists to assess and evaluate the vegetation type of an given ecosystem or a given community. To study the species diversity, it is helpful to understand the community composition, structure, change and development potential, and ultimately may lead to the conservation of the biological diversity. And biological spectrum expresses both life form distribution in a flora and the phytoclimate under which the prevailing life forms are evolved. Now, dealing with the characters used in community structure, or which what would be simply called as ecological analysis. For instance, if we say that a particular species is dominant in a community or particular population is a dominant in that community, what may be the possible characters on what basis that we have come to the conclusion that this particular species or particular population is the dominant one in that community. Therefore, there are few categories, few characters which can be used to categorize these characters, and we have generally divided these characters into analytical characters and synthetic characters. Analytical characters are those characters which can be expressed in terms of quantitative terms, which include like density, frequency, basal area, cover, and abundance. And on the basis of these analytical characters, some characters can be synthesized from these, which include important value index of that community, which include biodiversity index of that community, or what is the similarity index of that different communities, and these characters can be synthesized from these basic analytical characters. And there are different methods to calculate these analytical and synthetical characters, and one of them is the quadrant method. Quadrant is the name given to a sampling unit. It's an of a definite size, and it can be vary in shape. It can be circular, rectangular, or square, depending what is our objective to study. Since the assemblage of plants in a community is largely heterogeneous, the determination of the size of the quadrant and the number of the quadrants to be studied is a prerequisite condition in this case. And how to plot a quadrant, or what is the procedure to for a quadrant? A piece of string with three nails forms the L shape in the field. Using another piece of string and nails differentiates an area of 10 by 10 square centimeter. This is the initial first step while plotting a quadrant in the field. Now we note down the species, what are the number of species that will come under this area of 10 by 10 square centimeter. And the second step is we keep on increasing. We keep on increasing, enlarge the size from 10 by 10 to 20 by 20 square centimeter size of the coordinate, and we record all the additional species which have come up after first quarter, that was 10 by 10 square centimeters. And similar, we keep on enlarging the area. Then we go for 30 by 30. Sorry. Sorry. Yeah. Then we keep on enlarging the area of that quadrant until there's a time when there is no additional species is added in the area. Then that is the perfect size for the quadrant. Then we plot the number of species on the y-axis against the area of the on the x-axis. The point at which the curve start flattens up represents the minimum area of the quadrant required for the sampling in that particular field. And we usually call this method as species area curve method for calculating the area of the quadrant. Now, the second step is the how many number of quadrants should we plot. It depends upon the total area uh, of the field or the study area. We can go for 10% of the total area. We can go for 5% or 1%. It all depends upon the available resources we have at that time. And this is one of the students drawing the quadrats. On the right hand side, it is the quadrat for the shrub plants or grasses, and on the left hand side, it is the plot for trees. Uh, Following parameters can be studied under these florist diversity analysis or vegetation analysis or phytozoological studies in any given area. These include analytical characters like density, frequency, and basal area. And from these analytical characters, some synthetic characters like important value index, Shannon-Weaver diversity function, or Shannon diversity index can be calculated, or similarity index can be calculated. The term density represents the numerical strength of the species in a community. Density represents density
Dr. Asif sir, this or there is no audio and the slides are also not visible. Sir, you can just stop sharing and you can rejoin, sir. Participants are requested to stay back. Please bear with us. There is some technical issue. It will be resolved very soon.
हेलो।
हाँ हेलो डॉक्टर साहब, ये आप फिर से ये
सॉरी सर,
स्क्रीन
सॉरी डिस्कनेक्ट हो गया था, सॉरी मेरे को पता नहीं चला।
yeah, I told the participants to stay back. There was some technical issue. Click on the presentation. Yes, full screen mode.
हाँ,
But the slides are visible. Yes,
हो
just
गया
a
सर।
slide, one or two slides back. हाँ बस this slide, you were on this particular slide.
ओके सर, ओके सॉरी सर, ओके आई एम कनेक्टेड नाउ। Okay, so the different characters which are to be studied under the vegetation analysis include density, frequency, and basal area, and these three are the analytical characters. While as IVI, Shannon Diversity Index, or Similarity Index, these include the synthetic characters and can be calculated. These synthetic characters can be calculated on the basis of the analytical characters like density, frequency, and basal area. And the term density represents the numerical strength of a species in a particular community. And the density represents the number of individuals per unit of the area. And the density and the frequency are the prime important factors or characters that determine the community structure. So the procedure of calculating them is to lay the required number of quadrant of desired size and count each individual of each species in each quadrant. So the record can be the record the data can be recorded in the following manner, in which one column is of plant species and number of individuals per quadrant. Let there be let us assume there will be four quadrants, 1, 2, 3, 4, and then the next column will be the total number of individuals of all the quadrants what we have studied, and the total number of quadrants in which the species is present, and total number of quadrants studied, and then we will come up with the population density and the frequency percentage. And the density can be calculated by total number of individuals of a particular species in all the quadrants and divide by total number of quadrants studied, and it will give us density per unit of the area that is of the quadrant size. And similarly, we can calculate the density into from that area. We can calculate the density per unit, per hectare, or per acre, whatever is our objective. And the second character is the frequency. Frequency is the occurrence of a particular species. It will be calculated by the number of quadrants in which the species occurred divided by the number of quadrants studied, multiplied by 100. It gives the occurrence of a particular species in a community. For example, if we have four quadrants and a species is present only in two, then the frequency will come up 2 divided by 4 multiplied by 100. That means the 50% will be the occurrence of that particular species. And the basal area is the actual area of the ground or land that is covered by the tree or a plant. And in case of a tree, the basal area can be calculated by pi d square divided by 4, where d is the diameter, and the d diameter for the trees will be calculated at the breast height from the ground level, while as the diameter for the shrubs and the herbs will be calculated at the basal, at the ground level of the plant. So on the basis of these, there a separate compilation of the data is done to calculate the relative density, relative frequency, and the relative dominance of that particular species. The relative dominance will be calculated by dividing the density of a particular species divided by the total density of all the species multiplied by 100, while as the relative frequency will be calculated by by dividing the frequency of a particular species divided by total frequency of all the species multiplied by 100, and dominance, relative dominance, can be calculated by dividing the total basal area of a single species divided by total basal area of all the species multiplied by 100. These all relative density, relative frequency, and relative dominance of all the species when summed up will be the 100 for relative density, 100 for frequency, and 100 for relative dominance. And on the basis of these analytical characters, a synthetic character, that is important value index, will be calculated. If the important value index gives the importance of a particular species in a particular community, and can be calculated. Important value index of a particular species can be calculated by adding up its relative density, its relative frequency, and its relative basal area. And when we come up the summation of all the important values of the all species, it should come up with at 300 for all the communities. And similar synthetic characters like Shannon Weaver diversity index and similarity index can be calculated from this IVI of particular community. And the Shannon diversity index will give us then density of index of a particular community in that area. And similarly, the similarity index will give us the similarity between two communities which we are comparing. And the another character is the biological spectrum. Biological spectrum expresses both the life form distribution in a flora and protoclimate under which the prevailing life forms are evolved. The biological spectrum is prepared based on the percentage species composition in each life form. The growth form is mainly determined by the direction and extent of the growth of any plant. Uh, for example, trees, there is a large woody perennial with a single well-defined bole and a well-defined crown. A tree is usually more than 6 meters in height, while as a shrub is a woody perennial plant having a persistent stem and habitat of branching from its base. A shrub is usually not more than 6 meters in height, while as the herb is a plant with a stem is tender and green and its height is rarely more than 1 meters. And the climbers are those plants which need support to grow. And these are the rhinorrheous life forms which can be classified. Community can be classified in this form. That like therophytes are those animals which complete their life cycle from seed to seed in a single year and can survive any adverse climate as seed. Geophytes are the perennial plants whose bud is protected from freezing and drying by burying in the ground in form of a bulb or a rhizome. Hydrophytes are those plants which are grown in water and the bud remains below the surface of water and the above ground part dies at the end of the each growing season. Hermicraphites are the herbaceous perennials in which the aerial portion of the plant dies at the end of the growing season, leaving growing bud or at or just beneath the ground surface. Bryophytes are the shrubs with shoots or buds on the ground surface or approximately 25 meters above the surface, whereas phanerophytes are tall, woody, or herbaceous perennials with buds more than 50 meters above the soil surface. This can be dioecious trees and shrubs. And phanerophytes are again classified on the basis of height into megaphanerophytes, mesophanerophytes, and nanophanerophytes. And the biological spectrum for each class can be calculated by dividing the number of species falling in a particular life form class, divided by total number of species in that community, multiplied by 100. Similarly, on the basis of the leaf size, because the leaf size knowledge gives us the understand, the leaf size knowledge helps us to understand the physiological processes of the plant and the plant community, and is useful in classifying the association, and can be categorized. Different plants can be categorized on the basis of this leaf area into different types of the leaf size classes, and each size can be calculated by the number of species falling in a particular leaf size class divided by total number of species in that community multiplied by 100. Now, the second part of my presentation is on conservation prioritization of communities and habitat. After calculating the IVI of a particular community, if the IVI of a particular species in a particular community is more than 50%, a 50%, then we name that community on basis of that single tree species. For example, we have teak forests, we have sal forests, we have deodar forests. That means the 50% of the IVA of that forest is based on that particular species. So we name that forest on basis of that single tree. Whereas if there are two species who contribute about 50 or more than 50% of their or IVI, like we have fir spruce forests. That means these two species, fir and spruce, contribute about 50 or more than 50% of the uh, IVI in the community. That is why that forest is known as fir spruce forests. While if there are many species that add up and make the 50% or more than 50% of the IVA, then we name that plant community as mixed forest community. Similarly, on the basis of habitat, identification of habitat is done. Sites having close canopy with high percentage of moisture and high percentage of humus will be considered as moist habitats. Whereas if they are dry and there is less moisture, we call them as dry habitats. Similarly, if the area is more than 60% boulders, we call that boulder habitats. And if there is high anthropologic pressure on the any habitat, we call it as uh, degraded habitat. And the prioritization of the habitat and communities is done on using these parameters. First one is the species richness. Species richness gives the total number of species in a particular community. Economically important species, a structured interview is being formed, and the older people or village people are being consulted, and value of each plant and use value of each plant is noted from them. Native native are those species which are origin or they are first reported from the Himalayan region, then we call them as native of the Himalayas. Endemic are those plants which are restricted to Indian Himalayan region only, then we call them as endemic to the Himalayan region. Threatened plants we can calculate also, or we can use any version of the IUCN to know the threat categorize of that plant. Altitude is the altitudinal distribution of a particular plant. Site representation is the how many sites represent that particular species, and habitat is the habitat number, how many habitats include that particular species. Yeah. Then there are criteria for categorizing the prioritization of habitat and communities. It is on the basis of the score. Score is given from 2 to 10. 10 on the basis of richness, habitat, economically important species, native, endemism, threat species, site representation, and altitudinal range of species. The score is being given and is being summed up. For example, 10 score is given to that community which is having more than 50 number of species and is present only in one habitat, and the economic important species are more than 45% in the community. Native species are more than 45% in the community. Endemic are 40%, more than 40% in that community. Threatened plants are more than 40% in that community, and it is represented one by one site only, and altitudinal range distribution between the species is only 200. Then 10 value for each of the parameter is given. And similarly, the 8 value, 2, 6 value, 4 value, and 2 value is given for particular parameter in each community. For example, uh, this figure shows the family-wide distribution from the Gandharbal. One of the students have done it, and they have recorded that Astraceae family is the most dominating family in the particular region, followed by the Rosaceae and other leguminaceae plants. Similarly, this is the family-wise species composition in urban green spaces of Srinagar. Srinagar has also reported the Astraceae as dominant family in the area. Similarly, the growth form distribution in the Gandharbal was done, which has shown that about 59% of this total species out of the, I think, 115 species were recorded from the area, and 59 were the herbs, which contribute about 51% of the total percentage of the total community. While as trees were 29, shrubs were 22, subshrubs were 2, climbers were 2, and one form was observed from there. Then there was a comparison between because the Gandharbal area of the Kashmir includes both urban and non-urban parts. Then comparison was done between the urban and non-urban part of that district, and it was found that 59 herbs were found in the urban part, while as 43 were found in the non-urban areas of the Gandarbal district of J&K. Similarly, trees, 27 trees were found in the urban areas, while as 17 were there in non-urban parts. Similarly, shrubs, 21 shrubs were recorded from urban part, while as 16 were recorded from the non-urban areas. And climbers were two, they were only reported from the urban. Sub shrubs were those shrubs, two were from urban, while as one was from non-urban areas of the Gandarbal. Similarly, this nativity and endemism of flora of Gandharbal district was done. It showed that 101 out of 115 trees, 101 plants were non-native. Only two were those species which were native and endemic to the region, while as native plants were 12 in the region. Similarly, the diversity index was calculated for different areas of the Gandarbal district of Jammu and Kashmir, and it shows Dadarama having maximum diversity of 1.53, while as similarly the diversity indices of the non-urban of parts of the Gandarbal district was done, and it showed that the Tulmula having the highest biological diversity of 1.49. Similarly, the distribution of different life forms was done in the Gandarbal district of Jammu and Kashmir. It shown mega phanerophytes contributing the maximum 30, that is 34.78% of the total vegetation in the area, followed by uh, phanerophytes, nanophanerophytes, that is 17.39%. Similarly, the life forms in urban green spaces of Srinagar was done. The maximum was shown by threophytes, which was 42, uh, and followed by megaphanerophytes, that was 19. Representation of leaf types in the Gandarbal, 70 out of the 115 plants, 76 showed the simple leaves, and followed by compound leaf was found by 23, and needles were found in 6 plants. Similarly, the distribution of growth forms, leaf size, and leaf lamina was done in Srinagar also. 53 trees were found recorded from the Srinagar urban green spaces, followed by 11 shrubs, and herbs were 53 and trees were 37. Similarly, different leaf sizes were also recorded. The distribution of plant species in various habitats of Gandarbal was done. It showed that the 56 was found in park areas or urban areas, was having the maximum plant species richness in the area. Similarly, the biological spectrum of all the life forms was compared with the Reinecke's normal spectrum, and it was seen that maximum deviation from the Reinecke's normal spectrum was 34.78 in case of threophytes. That was 21.78% was deviation from the normal biological spectrum. It was then the prioritization of the plant community for conservation using different parameters was done, and it showed that the Platanus orientalis community showed the maximum conservation prioritization index with 46 as number. It was represented by one habitat. It was recorded at two sites, and the altitude, initial altitude was 1588 to 1618, and species richness was 57, and native species were 3 only, endemic were 2, economically important were 6. These were the this is the prioritization in of different plant communities in the Srinagar. This was the prioritization of habitats. So the riverine habitat showed the maximum conservation priority index with 40 value, followed by 36 was in shady areas, and near settlement areas it was also 36, and garden areas it was 32, roadside plantations were 32, and degraded were 28. Similarly, prioritization of the plant communities and conservation for different parameters were done for Gandarbal also. The maximum CPI, that is Conservation Prioritization Index, was calculated for the community Asculus indica Juglans regia community. Similar values were also obtained for Juglans regia elenthus community and Platinus orientalis plant community with 46 values, which was followed by Asculus indica community, that was 44 value was there. This shows the graphical representation of the same data. This is the prioritization of the habitat for conservation using different parameters. It showed that the uh, maximum CPI, that is Conservation Prioritization Index, was obtained for urban areas, green, that is garden areas, urban green space areas, with the value of 40. And this is the graphical representation of this only. And these are the species which were found in the Ganderbal district of the Jammu and Kashmir. For agroforestry purposes, Salix was there, different Salix species were there. Poplar is there, Almas was there, Rubinia was there, Elenthus, Escula, Esculus indica, Morus alba, and some conifers were also reported from the steady area. And similarly, the fruit trees, Prunica, Ganatum prunica, Prunus, Juglans regia, Ficus, these were reported from the area. And these were the crop species found in the area. And grasses were also there, Trifolium, Amaranthus, Bromus, Lolium. Similarly, the species in found in Srinagar were there. This is the table showing with their biological spectrum in Srinagar. Thank you. This was all from me. Any questions?
Thank you, Dr. Saab, for a very nice and I believe I would say a very practical aspects also touched upon. You explained everything.